Everyone, in this video we are going to learn about polymerase chain reaction PCR technique. Myself Sri Tallam, lecturer in zoology, DK Government College for Women, Autonomous, Nellore. So what is this PCR? Polymerase chain reaction is a method widely used to rapidly make millions to billion copies of specific DNA samples. So this laboratory technique generates large quantities of DNA. This PCR targets and amplifies a specific region of DNA strand. So allowing scientists to take a very small sample of DNA and amplify to a large enough so that we can study in detail about this DNA. So it is an in vitro technique to generate large quantities of specified DNA. This PCR technique works in the similar fashion like a photocopier or a Xerox machine through which we can multiply or copy number of copies within a short period. So coming to the history of this PCR machine, Carrie B. Mullis, an American biochemist, developed PCR technique in 1983 and was awarded Nobel Prize for Chemistry in 1993. Later on, the other important discovery was initially done by Thomas Brock, who discovered Thermus aquaticus, a heat stable bacteria which is usually lives in hot springs of Yellowstone National Park in America. Later on, from this Thermus aquaticus bacteria, Cetus Corporation scientists isolated thermostable tag polymerase, and this discovery has revolutionized the PCR technique, we can say that. The PCR machine is popularly called as thermocycler. Before the development of PCR, methods to amplify or multiply DNA were time consuming and labor intensive. So in this picture you can see the thermocycler machine. Now coming to the principle of PCR, the principle of PCR is based on enzymatic replication of nucleic acids. PCR involves the use of primer mediated enzymes for amplification of DNA. DNA polymerase synthesizes new strands of DNA complementary to template DNA. The DNA polymerase can add a nucleotide to the pre-existing 3-OH group only, therefore a primer is required. Coming to the requirements of PCR technique, we require a DNA template or target DNA that contains the DNA to amplify, which usually varies from 100 to 35,000 base pairs in length. And tag polymerase, that is heat stable polymerase, commonly used. So and two primers around 20 to 30 nucleotides in length, and that are complementary to the 3' ends of each sense and anti-sense strands of DNA target. And buffer solution which provides a chemical environment for optimum activity and stability of DNA polymerase. Bivalent ions like magnesium ions and monovalent cations like potassium ions are also required for this technique. The reaction is commonly carried out in a volume of 100 to 200 microliters in small reaction tubes of measurement 0.2 to 0.5 ml volume in a thermocycler machine. This thermocycler machine heats and cools the reaction tube to achieve the temperature required at each step of the reaction. So this thermocycler, in this picture you can see in this PCR tube, so all the components are mixed and placed in the thermocycler. So and the PCR cycle is carried out, which mainly contains 3 steps: denaturation, annealing and extension. So one cycle contains 3 steps, and in such a way around 20 to 30 cycles are carried out to form a required amount of DNA. So coming to the steps involved in polymerase chain reaction, step 1 is denaturation. Denaturation occurs when the reaction mixture which is placed in the reaction tubes is heated to 94 degrees Celsius for about 1 minute. So this temperature breaks the hydrogen bonds between the 2 strands of template DNA and converts into single stranded DNA. The single strand now acts as a template for the production of new strands of DNA. The temperature should be provided for a long time to ensure separation of 2 strands. So in this picture you can see the DNA strand is exposed to 94 degrees where the denaturation occurs and the two strands are separated. So, next one is annealing or renaturation. The reaction temperature is lowered to 54 to 60 degrees for around 1 minute. Here in these steps, the primers are annealed or bind to the complementary sequences on the template DNA. Primers are single strands of DNA around 20 to 30 bases in length. They serve as a starting point for synthesis of DNA. The two separated strands run in the opposite direction and consequently there are two primers necessary, a forward primer and a reverse primer. So, in this step we can see this annealing, the primers are binding to the separated DNA strands. So, next one is elongation step. At this step the temperature is raised to 74 degrees for 2 minutes. The bases are added to the 3' end of the primer by tag polymerase enzyme. This elongates the DNA in 5' to 3' direction. The DNA polymerases adds about 1000 base pairs per minute under optimum condition. So, as already we know, tag polymerase can tolerate very high temperature. It attaches to the primer and adds DNA bases to single strand. As a result, the double stranded DNA molecule is obtained. The three steps, denaturation, annealing and elongation steps, are repeated 20 to 40 times in order to obtain number of sequence of DNA of interest in a very short period. So, in this picture you can see one PCR cycle includes denaturation, annealing and extension. So, this picture also shows, so first we have denatured the double stranded DNA, along with we have added primers and DNTPs. So, in the denaturation step the two strands are separated, and annealing step the primer is annealed to the separated strands, and extension the DNA tag polymerase synthesizes, adds the nucleotides in direction. So, that is the elongation, and once again the same steps 1, 2, 3 denaturation, annealing and extension are repeated 20 to 40 times to get a required amount of DNA within a short period. So, in this picture we can see first cycle 4 copies are formed 2 to the power of 2, and second cycle 2 to the power of 3 8 copies are formed, and third cycle 16 and 32 copies. So, when we reach 2 to the power of 36 cycle, we get a 68 billion copies of DNA. So, there is an exponential amplification of doubling of DNA. So, in this picture you can see the temperature profile of PCR. A graph is drawn versus time versus temperature. So, the denaturation is done for 1 minute and annealing is done for 1 minute and extension is done at 74 degrees for 2 minutes. So, coming to the main components required, here DNA polymerase, the availability of this thermostable DNA polymerase enzyme isolated from the thermophilic bacteria or thermostable bacteria Thermus aquaticus founded in hot springs of Yellowstone National Park provided the means to automate the reaction. It has this particular enzyme has a temperature optimum of 72 degrees and survives prolonged exposure to temperature as high as 96 degrees, and so is still active after each denaturation steps. Tag polymerase lacks 3' to 5' proofreading exonuclease activity. Other polymerases are also found. TMA polymerase isolated from Thermotega marittima and PF polymerase from Pyrococcus furiosus. Nowadays, recombinantly developed tag polymerases or DNA polymerases are also being used in the PCR technique. So, and other very much important step is annealing temperature. Very important, since the success and the specificity of PCR depend on it, because DNA hybridization is a temperature dependent process. If annealing temperature is too high, pairing between the primer and template will not take place, then PCR will be a failure. Ideal annealing temperature must be low enough to enable hybridization between primer and template, but not enough to prevent amplification of non-target sites. So the annealing temperature should be usually 1 to 2 degrees or 5 degrees lower than melting temperature of template primer complex. So what is this melting temperature? Temperature at which two strands of DNA duplex dissociate. It can be determined experimentally or calculated from the below formula. Tm is equal to 4 into G plus C plus 2 into A plus D degrees Celsius. So here G, guanine, cytosine, adenine and thymine. So suppose we take a primer sequence containing this 4 nucleotide containing 3 guanines, 3 cytosine, 4 adenines and 4 thymines. We can calculate the melting temperature by using the formula, and it can be calculated as a 40 degrees Celsius. So we have to calculate the melting temperature also primer, and we have to set the annealing temperature, perfect annealing temperature, which is very important for PCR. So coming to the applications of PCR, this PCR is used in medicine for testing genetic disease mutations, monitoring gene in gene therapy, detection of cancer and viral infections. Nowadays COVID infections are identified by the RT-PCR technique. Forensic science, it is used as a tool in genetic fingerprinting. So we can identify the criminals from millions of people. We can solve the paternary issues, and research and genetics also. We can compare the genome of two organisms in genomic studies. In the phylogenetic analysis of DNA from any source such as fossils, analysis of gene expression and gene mapping. So all these are the applications of PCR technique.